Hello everybody, we're going to be talking about our topic of the week this week, which is Punnett squares. Before we get into it though, I'd like to do just a quick review of what we had been talking about last week, and in particular that family. So in this family, we had a trait of interest, and that was albinism. I got a lot of really great questions that I wanted to kind of save for next week when we talk a little bit more about what it is and how it happens. But just to be very clear, there is a colloquial term, albino, which is referring to the same group of people. However, I will always refer to it as people with albinism because they are people first. They're not just their characteristics. And that's how I would like you to refer to it as well. So that's our trait of interest. There are two phenotypes presented in this family. Remember, two different physical presentations of their genes. We have some parents who don't have albinism, and then we have children with albinism. We decided last time I talked to you that albinism was a recessive trait, and we kind of figured that out because a dominant trait is something that you only need one version of in order to show that, whereas a recessive trait is something where you need two of that trait to show it. So the only possible way that these two parents could have and who don't have albinism could have a child with albinism is if they're somehow able to be a carrier. They carry that trait for albinism with them. And we'll kind of dig a little bit deeper into that later. And you could, so let's just sit tight. So what are the genotypes in this family? So we know the phenotypes. We can figure that out pretty easily just by looking at them. But thinking about their genotypes is a little bit more work. So the only way to show a recessive trait is to have two copies of that trait, aka they are homozygous recessive. So we knew right away what the genotypes for the children were, and we decided to use M's. I'm just trying to keep it consistent with last week. So the children are homozygous recessive, or two lowercase M's, or two recessive alleles. We know that, what's her face had to get these through her parents, and her parents go through meiosis just like all other people, and they make sperm and egg cells, and they pass on one half of their information. So it doesn't matter if this is like the one from the mom or this is the one from the dad. It doesn't matter because they're both exactly the same. So both parents had to have carried a lowercase m, or a recessive allele for albinism. However, we know by looking at them that they don't have albinism, so they must also be carrying the dominant allele for melanin production, which would make both of the parents heterozygous for that trait, meaning that they have two different copies of alleles, two different things. They have a dominant and a recessive. Now, if we're thinking about this other brother, we actually don't know what's going on with him because there are two, we know that he produces melanin, but there's two different ways that, two different combinations of alleles that would result in the same phenotype. So unless we had genetic testing on the brother, we won't really know his genotype, because it's possible that he has two uppercase copies or two dominant copies, or he has one of each, just like his parents. But we do know that he doesn't have this combination, because that would mean that he has albinism, and we know that he doesn't have that. So we know he has one dominant allele, we just don't know about that second one. Okay, this is a great place to pause and take a look at the vocabulary if you're feeling a little bit confused by it or a little bit unsure. This is like a great place for you to check in and pause. All right. They're expecting! How exciting! The parents are expecting a fourth child, and they're wondering about the likelihood or the probability that their next child will have albinism. So, how are we going to figure this out? We are going to be using a Punnett square. And a Punnett square predicts the probability of an offspring inheriting a particular trait by analyzing the possible combinations of sperm and eggs. So this is a tool that you can use in genetics, and you likely have used it in middle school, but maybe you haven't. And I really like this drawing because it's super clear about what's going on. So this is the male parent's genotype. Okay, this is the genes that this male parent has. They're heterozygous, and we know that male individuals make sperm cells that carry half the information. So there's one half, or one outcome, is that the father donates a dominant allele, or the other possible outcome is that he donates a recessive allele. So we have two sperm cells with half the number of letters because it's half the number of information. Same with the mom, the female parent is donating only half of the information. So these little pictures are the egg cells. So when these, I'm like pointing at it with here, but you can't see that. But when the sperm cell and the egg cell combine, that's the baby, boom. And this baby would have two dominant alleles for whatever this trait is that we're looking at. But there are lots of other combinations that these two parents could possibly produce. And so that's why we have these single letters on the outside of the box, because they're representing the sperm cells or the egg cells. It doesn't matter if you put the the male parent up top or the male parent on the side. It doesn't really matter at all, just so long as they're going on the left and the top and not on the right and the bottom. So we've got single letter that will come together to form this double letter combo, and that's a possible outcome. Okay, so in this example, totally unrelated, we've got eye color. Many of us have heard that blue eyes is a recessive trait. So we have a mom whose phenotype is blue eyes. You can tell because we can see her eyes. And her genotype is homozygous recessive. Remember, the only way to show a recessive trait is if you have two copies of that recessive trait. So she's lowercase b, so she could possibly give away only lowercase b's. That's all she's got, so that's all she's going to give. So we separate them because this represents an egg cell. This represents another egg cell. The dad's phenotype is brown eyes. We know that his genotype is heterozygous, so he has both dominant and recessive alleles, and so we're splitting them up over here. And mom has two alleles for blue eyes, dad has one allele for blue and one allele for brown. Remember, being heterozygous means that you're carrying something that you do not show. He does not have blue eyes, but he does carry an allele for blue eyes. So what are the probabilities that the offspring will have blue or brown eyes? So to find the probability, we just do some very simple math. We're going to do the number of outcomes of interest, the thing that you're looking for, out of the total number of outcomes. So we would divide the number of, say, blue outcomes over the total number of outcomes. So in this example, the probability of blue eyes is 2 out of 4 because there were two possible ways to make an offspring with blue eyes, and 2 out of 4 reduces to 50%. We usually give our answers in percent, not in fractions. And the probability for brown eyes is 2 out of 4, or 50%. Remember, all of these percentages should add up to 100%. And it doesn't matter that these brown-eyed offspring are heterozygous because we're only asking about the phenotype. This question is only asking will they have brown or blue. They're not asking what kinds of genotypes. That would be a different question. If they were asking what's the percentage of genotypes for blue eyes, this would still be the same because the genotype, lowercase b. But if they were asking what is the possible or the probability of an offspring with homozygous dominant brown eyes, the answer would be 0% because there aren't any outcomes that have homozygous dominant or two uppercase b's. So you have to think, what is this question asking me? Is the question asking me about the phenotypes, or is this question asking me about the genotypes? Because those might not always be the same answer. Okay, so Tay-Sachs, just for a new example, it's a degenerative disease that breaks down the nerve cells in the brain and I think your spinal cord, and it is incurable. It's really unfortunate. It happens rarely, but higher in certain populations compared to others. So a carrier, I just want to be really clear about these words, a carrier has the recessive allele, but does not express that trait. So only a person with two recessive alleles actually has that condition. Homozygous dominant, capital A, and heterozygous individuals, capital A, lowercase a, are healthy. They have no symptoms. They don't have like half the symptoms as someone who has a full case of Tay-Sachs. They just don't have it, or they don't have any of the symptoms whatsoever. So, kind of continuing this Tay-Sachs example, remember it's a recessive genetic condition. So, two dominant alleles, two capital T's, would be a healthy person, nothing to worry about. A person who is heterozygous would also be a healthy individual, nothing to worry about. However, they are a carrier, and if that carrier ends up having a child with another carrier, it's possible that they have a child with Tay-Sachs, or those two recessive alleles. So in this example, we have two parents that are carriers of the Tay-Sachs allele. They're heterozygous. So right away, we know their genotypes. Mom is capital T, lowercase t, and so is dad. And so we would go to fill out this Punnett square over here to determine the probabilities for their offspring. So we're putting, and like I said, it doesn't matter if you put the female or the male up here, it doesn't matter. And in this case, they're exactly the same, so super easy. So now we're going to fill out the inside by combining this letter with this letter. They meet in this square, so we should have them both in that square. But over here, we're combining this capital T with this lowercase t. So we're going to have a heterozygous individual. And then we're going to continue just dropping these letters into the box until we're done. Boom. So now we've completed our Punnett square. So the question is, what is the probability that the mother and father will have a child with Tay-Sachs? So the number of outcomes of interest are the kids with Tay-Sachs, and there's only this one because this is the only genotype that results in Tay-Sachs. So that would be 1/4, or 25%. The next question is asking about the probability that they will have a child that is a carrier of the Tay-Sachs allele. Well, two of these individuals carry that Tay-Sachs allele. So that'd be 2 out of 4, which would be 50%. And the probability that the mother and father will have a child that does not have Tay-Sachs and is not a carrier, that would be 25%. That's just this. This is the only one who doesn't have Tay-Sachs and is not a carrier. Okay, so we are going to be working with some Google Docs where I've just made some like functional Punnett square tables that you are going to be typing into, and they look only slightly different than a traditional kind of Punnett square. And so here is where we would be having our outcomes. And in these outside boxes is where you would put the single alleles. Like this would be like one sperm cell. So there should only be one letter in these outside box each. It's like one egg cell. And then the outcomes, which have two letters, would be in the middle part of the box. And this is just blacked out because you aren't typing in there. There's nothing that needs to go in there. So you're just going to be typing directly into those boxes in order to fill out these Punnett squares instead of doing them on paper. If you are still having a little bit of trouble, or you're feeling a little bit confused about these Punnett squares, how they work, or how to make them, I've linked some additional videos that I think are really helpful in explaining how Punnett squares help us understand genetics, and I'm going to link those into the assignment as well. So there's a video from TED Ed about pea plants and genetics and the work that Gregor Mendel did, who's really kind of like the founding father of genetics. You've probably heard of him if you've talked about genetics in middle school. And then there's another video with the Amoeba sisters, which I love. I think this one was about hamsters. So feel free to check those videos out if you, or maybe they're guinea pigs. Either way, feel free to check those videos out if you're needing a little bit more assistance, and let me know if you are struggling. You can show up to my office hours. I can help you fill out these Punnett squares if you'd like, or you can send me an email or a text. But thank you.